as he delivers the 48th inaugural lecture of Babcock University titled Genetic Improvement of African Indigenous Vegetable Crops, a Panacea to Food Security, Sustainable Health, and Poverty Alleviation in Nigeria. Let us welcome him to the podium.
Thank you very much, Mr. President, Vice Chancellor. Once again, Mr. President, Vice Chancellor, distinguished ladies and gentlemen, permit me to stand on the existing protocol to present the 40th inaugural lecture of Babcock University. I am humbled and very grateful to God for the privilege to present the 40th inaugural lecture of Babcock University, God's own university. Today's inaugural lecture will be the 7th to be presented in the School of Science and Technology and the 5th in the Department of Agriculture and Industrial Technology. I cannot sufficiently express my gratitude to God, who has been my strength in the journey so far in my academic career. In the past, I have heard many of the inaugural lecturers assert that an inaugural lecture is a due that an academic owes to the academia. As truthful as this assertion may be, I think that even my delivery today will not be enough to pay the debt I owe to the academia, much more to the master teacher, Jesus Christ. Therefore, for me, it is a platform to share my experiences, views, and consistent learning and gains from students, colleagues, and most of all, the master teacher. Hence, by presenting this lecture, I have not yet attained the pinnacle of knowledge, just as the Apostle Paul would say it. I am still striving to reach the mark. Mr. President, Vice Chancellor, sir, permit me to briefly share my unprecedented interest and journey into the field of plant breeding. As a young boy, I had nurtured my interest to become a medical missionary because I love medicine, and I strived to gain admission to study medicine. Unfortunately, my hopes were dashed, and I was offered to study botany. I can see one of my botany, 30 years ago we met, and it's good to see you here. So I was fortunate to be given to study botany in the University of Port Harcourt, and so we continued. I still had the intention to change over to medicine until it struck me when I had my first class in genetics. And that's when my professor and supervisor, Professor Bosa Okoli, opened my eyes to possibilities in plant breeding, in genetics. I was so overwhelmed when I discovered the possibilities of regenerating a whole new plant from just a single cell and other exploits in the manipulation of genes. My interest was aroused to pursue a career path in genetics and crop breeding. I began to long for more knowledge in genes, their interactions, manipulations, and the roles they play in crop character improvement, especially the African indigenous vegetable crops, where I am today. Ever since, I have been swimming in this endless field of learning, yet to get to the end. As a seeker of true knowledge, I have always aligned my quest for true knowledge along the background that all truths are God's truth, and that God is the origin of true knowledge. And on this backdrop, I will introduce the topic of the inaugural lecture for today, which says, Genetic Improvement of African Indigenous Vegetable Crops, a Panacea to Food Security, Sustainable Health, and Poverty Alleviation in Nigeria. We all have a common denominator here. These three things: food security, sustainable health, and poverty alleviation is a common denominator that all of us share while sitting here, because everyone must eat. We are interested in knowing how we will survive. Of course, someone must have eaten before coming here and is hoping to eat again. And that's why this issue is very important. Among others, These are the three major issues of global concern in recent times. These issues have been worsened due to the skyrocketing global population and the effect of climate change. In order to address this, the United Nations in 2015, September 25th, adopted seven Sustainable Development Goals, which we call the SDG goals, which are aimed at ending poverty. And this is supposed to be actualized by the year 2030. The three topmost of the SDG goals focuses on poverty alleviation, taming hunger, and provision of good health and well-being to the citizens of the earth. 7 years on the countdown, this dream still seems what? Elusive. FAO 1996 defined food security as a condition that exists when all people at all times have sufficient, safe, and nutritious food to meet their dietary needs and food preferences for an active and healthy life. FAO 2009 has also identified 4 major pillars of food security, and that is namely availability, access, utilization, and stability. The first and the fourth pillars will be comfortably addressed through the promotion and genetic improvement of the African indigenous vegetable crops. For me, sustainable food security is thus the enduring capacity of any nation to produce food sufficiently to sustain her citizens and effectively ensure that food is adequately distributed to all without any compromises in food quality throughout her existence. By this definition, all and every machinery that has a nexus to food production, distribution, marketing, processing, and storage is called to question. However, every food production begins first with the sourcing of elite materials that have the capacity to turn out optimum yield, and also with quality nutrient characteristics. Koteva 2021 ranked nations based on the Food Security Index. Nigeria came what? 97 out of 113 countries, and 16 in sub-Saharan Africa, below countries such as Ghana, Mali, Cameroon, South Africa, Niger, and Benin. That is quite appalling. However, the African indigenous vegetables have the capacity to fulfill these criteria if deliberate and concerted effort towards enhancing their innate genetic potential via strategic crop improvement program is pursued. With this augmenting other crop production factors, efficiency in food production will be guaranteed. Let's have an overview of what African indigenous vegetable crops are. Most of us are used to them. The likes of okazi, Cocorus olitorius, ebolo, amunututu, all those. I don't know where they are today. Well, African indigenous vegetable crops are crops which are locally distributed and cultivated by early indigenous settlers of most communities in Africa over a long period of time. Their natural habitat originates in Africa, Skippers 2000. And most of these crops were sourced from the wild, where they grew in their natural environment. Prota in 2010 listed approximately 8,600 plants used in sub-Saharan Africa. Figure 1 and Table 1 will show pictures of some of these crops. Perhaps you will identify them and a list of those crops and some of the functions or roles they perform. Mr. President, Vice Chancellor, sir, I can still reminisce about some of those visits to my grandmother in the village during my early years as a young boy, with great anticipation for her locally prepared cuisine. I cannot forget the sweet and slippery feels of the blend of okra and obogoro vegetable soup, garnished with local mushroom. Nor can I forget the exquisite taste of okazi and oziza leaves, especially for women after birth. I will always remember clearly the likes of ogo, otabazi, angara, oda, just to mention a few. These are very rare African vegetables that afforded me unforgettable experiences. Unfortunately, some of these indigenous vegetable crops are rarely seen in our meals today. Meanwhile, these were some of the natural foods that may have contributed to the well-being and longevity of our parental generations. Where are they? That's the question. Where are these crops that are of great value? Let's look at some of the challenges that are confronting the AIVs. First of all, there are several of them. One of them is genetic erosion. The advent of urbanization and upsurge in the population of communities in Africa in recent past has led to a high demand for food to meet up with the challenges of population explosion. And arising from this is the intensification and expansion of agriculture to deal with the problem. This has consequently led to the destruction of the natural reserves where these AIVCs are. There are other challenges that has to do with cultivation. AIVC cultivation in most African rural communities is still in the unsustainable level due to the limitations of indigenous skills in cultivation practices. These skills are still underdeveloped to derive the full potential benefits of these crops. There is need to explore all the planting methods in crop cultivation and thus determine the best for the AIVCs. Poor viable seeds, a significant proportion of them produce seeds that experience dormancy. For instance, Cocorus olitorius and Ebelmuscus cali. Also Natium africanum. Some of them are even produce seeds that are not viable and can only be propagated vegetatively. Natium africanum, for instance. Some others find it difficult surviving in environments outside their natural environment and therefore will require intensive domestication programs. Low yield. Due to the limited research activities on the AIV crops, their yield is significantly limited. Their confinement to the natural environment has kept them obscured to possibilities for genetic improvement. The low yielding characteristics has conferred on them a limitation of meeting up with the increasing demand for food for national and global needs. The next challenge is competition. This is very crucial because there is a misconception that the exotic improved vegetable crops, such as kale, celery, cabbage, cucumber, lettuce, and others, are superior in nutritional value and have deprived the AIVCs their pride of place. And hardly would you find in the shelves of most vegetable sellers, even in our Babcock supermarket, hardly would you find such crops such as Vennonia amygdalina, that's bitter leaf, Piper guyanensis, Oziza, you have Solanum and Guivi, you don't find such crops there. Even when they are available, they are considered inferior and oftentimes command less cash. So farmers wouldn't want to go into it. And to solve some of these problems, there is need for these crops to undergo genetic improvement. Crop genetic improvement involves the manipulation of existing information present in the genetic code, the DNA, to direct the enhancement of crops in several characteristics, including yield, disease and pest resistance, improvement in agronomic traits, quality enhancement, and adaptability to biotic and abiotic stresses. Crop genetic improvement requires the accumulation and collection of genetic resources into gene banks, the evaluation and selection of genotypes with promising characters, and carefully using such genotypes in hybridization programs to produce new recombinant types with superior characters, which are faithfully multiplied for distribution to farmers to boost food security. Figure 2 shows a simple model of a plant breeding program, and that is quite self-explanatory. Accumulation of variability, naturally existing and artificially induced, and selection. That is very, very vital in any breeding program. And any mistake made there will determine progress or failure in breeding program, then evaluation, then multiplication and distribution. Genetic improvement of AIVCs and heritability. This is one other important aspect, and that's, as far as AIVCs are concerned, our selection is on the basis of what the heritability tells us, because our selection is on the basis of what we see. Heritability is a breeding concept used to describe the proportion of the total genotypic variance in the phenotypic variance, saying 2015. And in simple terms, it is the measure of the phenotypic variation that is accounted for by the gene in the population. The genotype is an expression of the genetic constitution of an organism, whereas the phenotype is the observable properties of the organism. And in the phenotype which the breeder considers in his selection, there are two things interacting. And what the breeder is interested in is the genotype, because that is what is heritable. And so the breeder needs to understand what is the proportion of the genotype in the phenotype that he sees. So if the proportion of the genotype is low, it means the environment is wielding more effect on the phenotype that is seen. And if he's choosing his crops on the basis of what he has seen, he may not make significant progress in improving that crop. And so he needs to understand the heritability component, which is the genetic component. This estimate of heritability of several characters in several AIVCs have been reported and deployed in the selection for possible improvement of some AIVCs. For instance, high heritability was reported in number of leaves per plant, plant height, leaf weight per plant, and total leaf weight in Cocorus solitorius. This was et al. 2018. In okra, Nwangburuka et al. 2012a. In Telferia occidentalis, Nwangburuka et al. 2014a. In soybean, Kumar et al. 2021, and have been deployed to aid selection for the improvement of these crops. At this time, I would like to look at the biblical foundation of crop improvement and faith integration. Mr. President, Vice Chancellor, sir, ladies and gentlemen, crop improvement has its origin from the beginning after creation by the Creator God. The command in the scriptures, be fruitful and increase in number, fill the earth, as found in Genesis chapter 1, verse 28, is a strong indication of God, the Almighty Creator's desire for the sustainability of his work of creation. In my own view, if you ask, I consider this command as the enabling power from the Creator God to allow his creatures to faithfully pass on God's genetic imprint peculiar to each species from generation to generation in order to maintain the continuity of each species, the Bible calls it kind, under the reproductive system. The means and wherewithal have been under divine guardians until sin came and altered the entire process of multiplication of species. In every act of multiplication or reproduction, the principle is to faithfully transfer or pass on the genetic or the gene component of a species from one generation to the other. Today, research and modern science have pushed mankind to explore beyond the limitations of such interspecific genetic exchanges to incorporate useful genetic materials from distant relatives and sometimes from unrelated species. Thus, DNA can be extracted, artificially synthesized, cut into fragments, rejoined differently, and sequenced at man's discretion. This great discovery of mankind has led to several manipulations of this complex molecule to bring about man's desired characteristics in species at will. This is the tool that powers the contemporary and modern crop improvement program. It is all about the transfer of DNA or gene from generation to generation. Therefore, Mr. President, sir, having laid this basic foundation, it would be convenient to present how genetic improvement of AIVCs could help in achieving sustainable food security in Nigeria. The prospects of genetic improvement of AIVCs for sustainable food security. Mr. President, sir, huge opportunities for genetic improvement still abound in the improvement of the AIVCs, adopting biotechnological approach. This approach provides solutions to overcoming the challenge of non-viable seeds and production of plant propagules, transfer of important genes between AIVCs irrespective of their genetic distances, and preservation of the AIVC germplasms ex situ instead of relegating them to their natural environments. I would like to propose a few contemporary genetic improvement approaches adopting biotechnology that can be explored to enhance the performance and quality of AIVCs. The first is somatic embryogenesis. Somatic embryogenesis is the regeneration of embryos from somatic cells, unlike the zygotic embryogenesis, which require diffusion. So in this case, we are eliminating sexual reproduction. This is an asexual form of regenerating propagules for AIVCs. The SE approach involves the culturing of a cell, group of cells or somatic tissues referred to as explants in an enhanced aseptic nutrient medium. These cells undergo a series of changes as they multiply in the medium, giving rise to several plantlets as end products. This approach has been considered by many researchers as a better option in the genetic improvement of most woody and horticultural plants, Vierbav et al. 2019. SE has also been applied in the propagation of guava, mango, papaya, among others, Singh et al. 2007, and can also be explored in the production of synthetic seeds for most AIVCs that cannot produce viable seeds, Rai et al. 2009. Figure 3 is a schematic description of the stages involved in the rapid multiplication of, for instance, guava using SE. By the way, I have a PhD student who is working on this. We are trying to develop an SE protocol in regenerating plantlets for tiger nut, which is also an AIV crop in the north. And the work is almost in its concluding stage. And very soon we'll be regenerating those plantlets. So the application of SE will ultimately enhance the rapid and readily availability of the AIV crops and also enhance their introduction. Embryo rescue. This is another useful tool in the hand of a plant breeder. In order to improve AIVs further in nutrient content and other useful agronomic characters, there is need to successfully hybridize distant relatives of AIVCs with distinct desirable characters to produce a better version of themselves. ER involves the recovery of fully developed hybrid embryos from distant interspecific or intergeneric species of plants after fertilization, which ordinarily would degenerate or abort due to incompatibility between the embryo and the endosperm of the ovary. Premanak et al. 2021. By this technology, AIVCs can be genetically improved for better performances using better characters present in their distant relatives. For instance, AIVs can now be improved qualitatively and quantitatively to combine enhanced yield with essential vitamins and proteins, as well as resistant to biotic and abiotic stresses. Molecular marker-assisted selection, MAS, for resistance to biotic and abiotic stresses. Mr. President, Vice Chancellor, ladies and gentlemen, the limitation of the conventional breeding as well as the constant evolution of the pathogens that plague the AIVCs have slowed down the pace of progress achieved over time, Afari Safar et al. 2012. However, the complementation of the molecular tool using marker-assisted selection, MAS, will likely speed up the rate of progress in the development of pest and disease-resistant varieties of AIVCs. Of course, in the conventional or contemporary plant breeding, we depend on what we see. And that's why we adopt heritability to serve as the index to measure the reliability or the efficiency of our selection. But when you use MAS, it augments because you are dealing with the gene, excluding all environmental conditions, which influences the phenotype. Molecular markers are distinct DNA sequences present in the genome of crop species, which can be used to probe the presence of specific desirable characters in crop, and therefore can assist breeders in selection of characters in crop improvement programs. The development of such molecular markers in AIVs to aid selection for their improvement is still at infancy, and that calls for great caution. Afarisafa et al. 2012. And a typical effort towards developing disease resistance in vegetable crops in Africa using MAS is the development of the bacterial wilt resistant tomato lines by AVRDC. Professor Denton will know this, in Arusha, Tanzania, and has been undergoing trials in Eastern and Southern Africa. Therefore, development of such molecular markers will boost progress in the development of AIVCs that are resistant to the varieties of pests and diseases, as well as tolerant to most biotic and abiotic stresses. Disease-free propagules from meristem culture. Mr. President, Vice Chancellor, sir, it is also possible to develop AIVCs that are disease-free for cultivation using an in vitro culture technique known as meristem culture. Ample scientific evidences have shown that cells located in the apical region of plants are totipotent and usually disease-free. Robert, 2013. Figure 4 shows a picture of plants indicating the areas that are where we have the apical meristem. We have at the shoot tip and at the root tip. Totipotency is the ability of cells to constantly divide, giving rise to cells which later differentiate into specialized tissues. This discovery has swung open the door to various possibilities in the development of various disease-free crops following meristem culture. Meristem culture is the growing of cells from the apical region of plants in an aseptic nutrient medium to produce several disease-free propagules. So with this, we'll be able to produce several AIVCs that are virus-free, that are disease-free. Figure 4 there shows the schematic or diagram showing the steps involved in tissue culture. You get the apical tissues, you surface sterilize it, then you culture them in the medium, and several activities take place which will lead to proliferation of several cells which will eventually differentiate to produce plantlets, and these plantlets will be acclimatized for the environment. Figure 5 shows a typical tissue culture lab. AIVCs in sustainable food security. This is where we are. And let's look at AIVCs, a genetic improvement of AIVCs for sustainable food security. There are more than 45,000 indigenous crop species in sub-Saharan Africa, and approximately 1,000 of them are consumed as leafy green vegetables and also constitute the majority of traditional African diets. Makela, 1994. Adeboye also reported about 24 of these species have been reported in Western Nigeria. Adeboye et al., 2005. Now the question is, if we have this wide variety of options, why are we limited to just a few? A bag of rice is 40,000 naira, and we are all crying. If we don't have rice, it means we will die. It will be a repetition of what happened in Ireland in the 1840s, when they depended solely on Irish potato. And you know, when the Irish potato plague struck, so many Irish people died, over 1 million, and over 1 million migrated to the United States. So if we have this wide range of options, why do we limit ourselves? Why can't we go back to what we used to have? Why can't you go back to crops like ebolo, like amonotutu, like okazi, and develop them? When I was presenting this, someone wrote to me and said, Syngenta will not what? They will not sponsor you. You know, they are one of the biotechnology companies. Because I want to talk about what? Our indigenous crops, which we have relegated. And we need to go back to them. And that's the whole essence of this. Let's cash in on this wide variety that we have and look at it. There is large variation among these crops. For instance, Nwangburuka et al. 2012 established significant genetic diversities in cultivated okra. Biswas also reported similar variation in kokoros. And other AIVCs have with huge interspecific genetic diversities include Telferia. Therefore, these large genetic diversities in AIVCs provide unlimited opportunities for us to do what? To improve these crops. Thus, I hereby propose that a deliberate and intensive research approach towards the utilization of the huge available genetic diversities within the AIVCs would undoubtedly promote what? Sustainable food security in Nigeria and Africa at large. Improvement of AIVCs for sustainable health. Studies have shown that countries where people retain diets rich in AIVCs are less likely to be affected by cardiovascular diseases, diabetes, and adverse consequences of nutrition transition Jones and Stapit, 2004. AIVCs are widely known to possess antioxidants, especially when eaten raw. This makes them potential natural remedies to most ailments and diseases common to local urban communities. They have come to be cheaper ways of dealing with cellular oxidative damage and used in treatment of cardiovascular diseases and cancers. Yusiku et al., 2010. And I want to buttress this because in a Proctor research, in a Proctor-funded research survey which we conducted, Denton et al. In 2010, we observed in Ogun, Oyo, and Lagos State, we observed that 90% of plant species in the study area had one or more medicinal values, apart from either being used as food and fodder. And most of the plants we discovered were plants like Cromonena odoratum, we have Cocorus olitorius, Ebemuscus esculentus, and Vennonia amygdalina. These were the crops that topped the research results that we had. So we should be very thankful that we have this to deal with not only our food problems, but our health problems. Therefore, I hereby propose an extensive genetic and chemical characterization of AIVCs in order to accurately generate robust data on their medicinal values and use in the treatment of common diseases associated to African communities. The table 2 there shows some of the list of the indigenous African vegetables and some of the diseases that they have been associated to. These are verified results. Improvement of AIVCs for poverty alleviation. Evidences from research shows that the average revenue from 3 selected premium AIV crops were $540 and $200 in Abidjan and Kampala, respectively. From our own observation in southwestern Nigeria, it reveals that average income from cultivation of Terferia occidentalis, that's Ugu, in a typical rainy and dry season are $1,738 and $1,891, respectively. Nwangburuka et al., 2018. Similar observation was reported by Adeboye, 2004, in southwestern Nigeria. He observed that Solanacea biafrae, an indigenous vegetable, fetched more income in the dry season compared to regular cultivated exotic species. Who says the exotic vegetable crops are more valuable if our indigenous species fetch more money? Other indigenous vegetables, such as C. argentea, A. cruentus, and S. macrocarpon, have been reported to yield higher revenue in southwestern Nigeria during dry season. Adeboye and Okpabodi, 2007. Therefore, it is thus proposed that a domestication of more of the AIVCs should serve as potential solution in poverty alleviation and improvement in farmer livelihood in an environmentally sustainable manner. The improvement of AIVCs for enhanced nutrition status. The highest undernutrition statistics is found in sub-Saharan Africa, with 1 in 3 people being chronically hungry, FAO 2008. Similarly, there is a strong nexus between lack of micronutrients and poor health, Saka et al., 2007. Several research efforts have identified AIVCs as important sources of nutrients in the African diet and the impact of their processing and cooking on nutrient content being excellent sources of vitamin A, B complex, C, and E, as well as iron. In comparison with cabbage, amaranth green has been shown to contain 57 times more vitamin A precursor, 13 times more iron, and 8 times more calcium. Yang and Kedin, 2012. Moringa has also falls in that same line. Therefore, AIVC represents a cheap but high quality source of nutrition for poor populations, especially where malnutrition is endemic. Nnamani et al., 2009. Table 2 will show you a display of some of the crops. I purposely arranged them. You will see the last 3 there are the exotic crops. Compare them with the ones on top in terms of energy, protein, calcium, and other nutrients, you will discover that our indigenous African vegetable crops are doing better. So why are we relegating them? Why do they not make the bulk of our food? And we are so proud to buy cabbage. We feel so good. We feel so good when we buy cucumber because we think, oh, this is a white man's food. That's what drives us. I think it's high time we start changing the narrative. My contribution to research, please don't be offended. My contributions to research in improvement of AIVCs. Mr. President, Vice Chancellor, sir, I have dedicated the greater part of my research life to understanding the genetic diversity and characterizing several indigenous leafy vegetables. I have evaluated and characterized okra accessions comprising of genotypes from the 5 geopolitical zones of Nigeria and a few exotic accessions using morphological and molecular tools. I observed huge genetic variations among our indigenous accessions, enough to effect significant improvement in yield and other agronomic characters in okra. Nwangburuka et al., 2012a. Similarly, I and my research colleagues have also characterized and reported huge genetic variations in other indigenous vegetable crops, such as Coccurus solitorius, Tephero occidentalis, Celosia argentea, Solanum anguivi, Lycopersicum esculentum, and Vigna unguiculata. Nwangburuka et al., Nwangburuka Dentin, and Dentin, Nwangburuka et al., 2014a, Dentin and Nwangburuka 2011, Ezekiel et al., 2011, Nwangburuka 2004. This information provides huge prospects for genetic improvement of these indigenous vegetable crops for food security and poverty alleviation. We have also conducted significant researches on the heritability of several agronomic traits of indigenous vegetables, on the heritability of agronomic and yield characters in okra and indigenous fruit vegetable. We observed high heritability and genetic advances in the yield characters, such as days to flowering, plant height at maturity, mature pod length, pod weight per plant, and number of branches per plant, which provides huge prospects for the genetic improvement in these attributes of okra crop through intensive selection. The high heritability observed, we suggest that these were controlled by additive genes. Nwangburuka et al., 2012a. Other crops we studied their heritability include Coccurus, T. occidentalis, and Solanum anguivi, Celosia argentea. Mr. President, Vice Chancellor, in order to aid conventional crop breeding and the application of biotechnology in crop improvement, we have characterized the chromosomes present in the cultivated okra. Our findings clustered okra genome into 8 groups on the basis of chromosome morphology. Significant proportion of the chromosomes were asymmetrical and ranged in length from 3 to 16 micron. Nwangburuka et al., 2011. This information is very useful in creating the genetic mapping of okra genome for improvement through biotechnology. We have also evaluated and characterized several indigenous okra accessions assembled in the gene bank of the National Institute for Horticultural Research, NIHORT, for stability and yield, comparing them with some elite exotic varieties. We observed and recommended 3 indigenous accessions, such as LD88-1-8-5-2, 47-4, and NH88-1-8-16-2. These 3 were best in yield and stability in the 4 different agroecological environments where we tested them. And we have also recommended them to farmers, believing that it will boost their economics. Mr. President, Vice Chancellor, sir, I and my research team worked to provide a sustainable solution for handling of poultry waste, as well as reducing the Herculean cost of inorganic fertilizer by developing an alternative to inorganic fertilizer. This project was funded by the Adventist Development and Relief Agency, ADRA. The technology, we called it the Arbuscular mycorrhiza fungi poultry manure, AMFPM fertilizer or biofertilizer. This biofertilizer has been used in field experiments with okra, amaranthus, ugu, Nwangburuka et al., 2012b, 2014b. The AMFPM technology was promoted and used to boost the cultivation capacity and yield of ugu leafy vegetable production amongst rural women farmers in 3 vegetable farming communities of Ogun State, namely Ikene, Odogbolu, and Okonwa. the women vegetable farming communities of over 300 women attested to the increased yield and higher revenue from ugu production using AMF-PM fertilizer. The AMF-PM competed favorably with the commonly used inorganic fertilizer to sustain yield, preserve soil health by boosting soil fertility and soil beneficial microbial population. Nwangburuka et al. 2018. We observed that the T. occidentalis farmers were willing to adopt this new soil fertility booster. However, Mr. President, sir, as impacting as this work is, we have been constrained by limited funding to initiate an upscaling of this AMF-PM project to have a wider impact. Plates 1, 2, 3, 4 are pictures of when we were discussing with the farmers after that's the post-trial. The first one was when we started, and the project is still pending. We just need funds to escalate it. We have also investigated and recommended optimum seed treatment with dry heat for C. olitorius, indigenous African leafy vegetable crop, to boost seedling emergence, germination vigor, and overcome the limitations of seed dormancy. Denton et al. 2013, we observed that an exposure of cocorus seeds to 120 degrees Celsius of dry heat for 5 minutes promoted optimum seed germination, emergence, and vigor. I collaborated with my research group members to conduct an assessment of the status and usability of stockpile soils during mining activities in South Africa. Ezekwoli et al. 2018. This was a South African National Research Foundation funded research. Our finding was that the stockpiling activity disturbed and influenced the abundance and variability of AMF in stockpiles. We also observed that duration of the stockpiling and the way in which the soil were stockpiled influenced the capacity of using the stockpile soils in land reclamation for agricultural use. This research recommended that soil stockpiling be conducted with minimal disturbances to the topsoil structure, and that the use of heavy equipment over topsoil stockpiles is minimized to prevent soil compaction, which is linked to AMF spore survival. The list is endless, but permit me to just say this before I do my recommendation. I have also collaborated in several research projects aimed at investigating food safety and identification of microbial diversity in food during processing in order to mitigate and guide against food contamination from mycotoxin. A few of such studies are the investigation of the mycotoxin profiling and bacteria communities during processing of different formulations of kunu, a traditional food beverage, Ezekiel et al. 2019, and bacterial species and mycotoxin contamination associated with locust bean, melon, and their fermented products in southwestern Nigeria, DDG et al. 2017. These studies reveal that fermentation processes of syrup foods reduce significantly the mycotoxins of the processed food product. However, caution must be exercised to ensure for proper hygiene during fermentation process, as this was implicated as possible sources of microbial contamination in food processing. Service to Babcock. I will leave this because my President Vice Chancellor has actually mentioned this when he was reading my citation. So let me leave that and just pick some of the recommendations before I round off. There should be an intensified effort by both the government and private sectors through research, exploration, and assemblage to rescue the available AIVCs in order to prevent further genetic erosion and extinction of the AIVCs. The government, too, and private institutions like Babcock University should invest in the establishment and maintenance of standard germplasms to accumulate, protect, and preserve the genotypes and the different genetic resources, particularly the AIVCs, to serve as repositories for future genetic improvement. And so NAGRAP should be funded because it's the center that manages projects like that. By the way, we have an MOU with NAGRAP, and we have a small germplasm here. I want to appeal on the Vice Chancellor to also look that way, so that we'll have a formidable germplasm. 3, a general evaluation and characterization of the vast array of the available AIVCs should be carefully embarked upon by researchers using molecular and morphological tools to ascertain and describe with accuracy the diversity and distinct characteristics of genotypes of AIVCs. This will generate robust data that will assist in the genetic improvement of crops. Let me go to 8. We can read the rest. I am proposing a deliberate collaboration between the indigenous communities who are more conversant with the values and uses of the AIVCs and the conventional plant scientists to promote identification, collection, and preservation of the endangered AIVCs, as well as an extraction and purification of useful bioactive substances from these plants. Finally, since biotechnological technique is very essential in attaining success in the genetic improvement of the AIVCs, researchers of AIV crops should undergo requisite training on biotechnological techniques to aid in the genetic manipulations required for crop improvement. Acknowledgement. I am forever grateful to Jehovah my God, the creator and keeper of my life, for his personal and careful ordering of my path as I navigate the academic arena. My sincere gratitude also goes to my late father, Mr. Nwangburuka, who envisioned this but did not see it. And to my dear mother, Mrs. Ruth Nwangburuka, who is present here. Both my father and my mother invested, they sacrificed so much to invest and inculcate values to us. We couldn't have had any better training. Thank you very much, Mom. We love you. I also would like to appreciate my mom and dad-in-law, that's late Chief Engineer Ayo and Engineer Ayo and Madam Rachel Adeyemo of blessed memory, for their care and love. In a special way, I want to thank my siblings, Mr. Nwangburuka and his family, my twin sister, who would have loved to be here, but she's there in the US, Mrs. Ugochi Njoko and her family, my brother who is here, Mr. Chidi and his family, my brother who is not here too, who would have loved to be here, Mr. Chikere and his family, my brother, Engineer Gozier, who is here, Engineer Hope, who is also here, and their families. You have constantly provided support and immense encouragement for me to forge ahead. May God bless you. I want to remember all other Nwangburuka family, Wanju, Dineya, Nwabara, my cousins, nieces, and nephews. I want to thank you for being the support that you have been. I'm also grateful to my in-laws, Mr. Adeniyi and his wife, Dr. Yewande, Mrs. Ogundiwi, Adeola and her husband, Dr. Benice Ogundiwi, Mrs. Lola and her family too. May the Lord continue to bless you for being the family that you have been. I'm grateful to the Babcock University administration, ably led by the indefatigable President Vice Chancellor Professor Ademola Stephen Tayo, for providing this platform for me to make this presentation. Sir, may God continue to guide you as you lead Babcock University to glory. Amen. I will not forget the imprints of these leaders, Professor Adekunle Alalade, Professor J.A. Kayode Makinde, Professor A.C. Omono, Professor Luke Onoha. Thank you very much, sir, for honoring my invitation. May God bless you all and your families. My appreciation and thanks goes to Professor Iheanyi Chukwu Okoro, my immediate past Senior Vice President Academics. Sir, you are here. I dressed like this for you two today. And Professor Philemon Amanze, who has been at my neck, my immediate boss. Sir, may God continue to bless you for the support and encouragement you have provided to me. I will always remember and appreciate the support provided by Professor Jacob Haliso, Professor A.D. Aino, Professor Jonathan Wosu, Dr. Sunday Audu, and Dr. Folorunsho Akonde, as well as Dr. Oladipupo Adebawo. I will also like to use this opportunity to acknowledge and thank the provost of colleges and the deans of schools at Babcock University. Your names are there. Many thanks to all the heads of departments in the School of Science and Technology. These people have been So wonderful. Actually, they made my work as a dean very easy. May God bless you, Professor Olajuigbe, Dr. Kajetan, Dr. Olarimoye, Professor Isaac Oyewole. My sincere thanks goes to all other members of the School of Science and Technology. Please, I will not be able to list all of them as they are there, and many that I couldn't list. Many thanks to Professor Sisimosu, Professor Awodele, the leaders of the academic planning and OIE. My thanks to the members of the think tank committee that worked with me in agricultural-related projects. Let me thank all of you there. I am immensely grateful to the inaugural lecture committee, Professor Grace Tayo, Professor Dora Kimboye, Emeritus Professor Michael Omolewa, Professor Gabriel Alegbele, Dr. Kola Yodele. You people did a great job in making this work what it is today. I owe a lot of gratitude to my dear colleagues in the Department of Agriculture and Industrial Technology, from the past to the present. Professor Denton, thank you very much. The past are there, and I'm listing them. They are all listed there, and the present. Please, I acknowledge all of you, and I thank you for making me the person that I am today. I appreciate my colleagues in the Research, Innovation, and International Cooperation, RIC, especially my dear friend and sister, the director, Professor Grace Tayo, and all the teams of RIC. They have been part of my success story. I appreciate Professor K.O. Ogunemo, the immediate past Vice Chancellor, Adventist Universities of West Africa, for giving me the opportunity to contribute as the Vice President, Academic Affairs, Adventist University of West Africa. I also appreciate Professor Godwin Aja for his support over the years. I wouldn't be anywhere today without the seed sown by my teachers. I want to remember especially Mrs. Huanko. She taught me maths when I was in secondary school. She was a Russian, and I can't remember how she grilled me. I want to thank her, even though she's not here. Professor Bosa Okoli, who actually initiated me into plant breeding and genetics. Doc, you know. So I want to thank him. I thought he would have been here. Professor Aifawole, another grilling machine that grilled me for my master's, and would have done it in PhD. But he gave me his son, Professor Kehinde Babatunde, who, I don't know if he's here. Is Professor Kehinde here? He's the DVC management in Funab. He said he would have been here, but they are having canceled visitation today. I want to thank them and many other friends from Funab. I'm also grateful to my friends who comforted me during my undergraduate days, Professor Matthew Wegu, Professor Gunji Johnny, Engineer Kosima, and several others. Many thanks to my colleagues in the class 1992 set. I'm really so grateful to my friend who came today. It's over 30 years that we saw last, and he made it. This is the first time I'm seeing him after 30 years. I want to thank you. Please pass my message to all other botany guys in Uniport. They are in different places doing well. Their names are there. I also want to thank my host at the Agricultural Research Council, Institute of Soil, Climate, and Water, Pretoria, Professor Adeleke Rashid, and the entire research team. We still work together. I want to thank them. I felt this would have been on Zoom for them to participate. I appreciate my friends in the Mushroom Development Foundation, ably led by Chitola Roberts and Amaka Roberts. The appreciation also goes to the General Commander, Director, and officers of the Dominion Chaplaincy Corps. To all my students, undergraduate and postgraduate here and abroad, I thank them for sharpening my sword for better efficiency. They were the fire that refined me. Each moment with you in a class, field, or laboratory brought me new experiences. Thanks so much. And with particular reference, I would like to thank Dr. Habib and Mrs. Adetutu, Stephen, who are here. They are my students. They have become my colleagues. I want to thank you for the support, even towards this program. Mr. Abiodun Deji, who is in the US, I also want to thank him for the support towards this program. Mr. Deuti and several others, I want to thank you for your support. I am eternally grateful to members of the Seventh-day Adventists. This is my spiritual family, Town Planning. I want to thank you for the nurture you have given me. Your prayers meant so much and still means a lot to me. I want to thank you, my pastor, Pastor Ajani is here, Pastor Lubayo Ajani, Pastor Olajide, my current pastor, Jacob, is also here. I want to thank you, my head elder, Elder Lawal Akinshola, and the entire elders and members of Seventh-day Adventist Church Town Planning. I want to appreciate all the Nation West District. I say thank you. My president, I just called you. My two presidents are here. You see, I have the spiritual support. The president of what? Ogun Conference, where I reside, Pastor Dr. Sunday Ogunsoya. He's here. Thank you very much, sir, for coming. And two of them are sitting together, too, and their wives. Thank you very much, sir, for coming. Pastor Dr. Ezekiel Oniloye. He is also my president in Oyo Conference, where I came from. They posted me to this place. I want to thank you. I'm still doing the work you sent me here to do. May God continue to bless and keep you. The Orion Class 2017, I want to thank you, Dr. Theodore, who was my co-sponsor, and the executives. Finally, I say a big thank you to my heartbeat and dearest wife, Mrs. Adekemi Esther Nwangburuka, who has been there for me all the time. She has stood by me, soaking up the stresses and pressures and never tired of me. Baby, you have been my constant support. And the wind beneath my wing, thank you for believing in me. She's my true friend and the best for me. To my dear son, Samuel Onyekachi Nwangburuka, I say thank you for your love, support, and understanding. I will always remember your sincere encouragement and prayers for Daddy's success. May God make you the man after his own heart. I also want to thank, where is Professor Ananaba Paul? I want to thank him because he's supposed to be in a tribunal. He told me he was going to cancel it, and he's here. I want to thank him. I thank Professor Afolami. Prof, I want to thank both of you for honoring me. I'm so glad. I'm so overwhelmed when I see faces here today. I cannot count or begin to mention all your names, but you have actually touched my heart, telling me how much you care. To everyone that has come here today, I want to thank so many other people, the committees that put this thing together. I want to thank Bro Ife, who has also been consistent. The committee, the women committee that worked on some of the, I want to thank you because I wouldn't call all the names to be able to do that, but I appreciate you all. All the members of School of Science and Technology, I thank you for your support. And to everyone that has come here today to honor me this day, may God honor you and your families. May the blessings of God follow everybody as they live here today. Amen. Can I just take the conclusion, sir? All right. In conclusion, I wish to submit that the steady global growth in human population resonates a deep concern for an integrated approach for a sustainable food production, management, and distribution system at all times. This is one way of achieving food security. Meanwhile, AIVCs could provide the huge underutilized natural food resources, which could conveniently complement the already existing food system. However, the deliberate ignorance and lack of awareness is gradually sidelining this huge resource of AIVCs into genetic erosion. Therefore, the urgent need to preserve the significantly depleted AIVCs for food security. Therefore, an intentional approach towards harnessing the huge diversity within the AIVCs through research efforts geared towards improving the genetic, nutritional, and adaptability of the AIVCs to the agroecological system may be a potential panacea to sustainable food security, health, and poverty alleviation in Nigeria. Mr. President, Vice Chancellor, sir, this is where I rest my case. Thank you, sir.
Please, you can have your seat. Ladies and gentlemen, you will all agree with me that within the matter of 58 minutes, because it started 2:36, he has successfully convinced me and many people here that we need to go back to our roots and ditch cabbage and go for okra and all sorts. He has called for rescue of African indigenous vegetable crops because in terms of nutritional value, they are better. In terms of medicinal value, they are better. I want to thank you very much for advocating for domestication and development of better cultivation protocols. Thank you very much for the wonderful presentation. And now, by the power conferred on me by the Babcock University Governing Council, I hereby officially admit you to the professorial chair as Professor of Plant Breeding and Genetics of Babcock University. With all his rights and privileges. Congratulations. As you all know, inaugural lecture is usually a solemn or very serious ceremony with little or fanfare within the hall. Therefore, to capture this very important ceremony, we have group pictures outside. But permit me to use the privilege of being the chair to invite the baby as a please, baby, you have to come forward. And Samuel, you have to come forward. And Mama, please get ready. After the family has taken the picture, Mama will join. And the rest of us will have our own outside. Thank you so much. Samuel, you have to help baby. Thank you very much. Madam Ruth Nwangburuka, please, Mom, you can come forward, ma'am. I think the Nwangburukas, I know you have a very large family. Please join Mama here. I'm seeing a lot of engineers here. Please come forward and join your siblings. The Nwangburukas are very tall people. Oh. Very good. Thank you. The last but not the least, the principal officers that are here and the members of the inaugural lecture planning committee too. I think my own baby should be here too. I think I've learned that language today. Well, I think I've seen the members of the inaugural lecture planning committee. Emeritus Professor Molewa, please. And a few others, please. You have been working very hard and we should recognize you. After this, all other pictures will be taken outside. And I think after that we have a cocktail party for the invited guests at the Dr. Ayodele. Okay, the others are not here. Okay. Thank you very much and God bless you all.
I invite Dr. Ayodeji Olarimoye for the closing prayer.
Let's pray. Our Father and our God in whom we trust, we thank you so much for the way in which you have allowed this program to go. We thank you for taking absolute control, for making it a beautiful event. We thank you for empowering the speaker of the day to address the matter. We want to thank you also for bringing everybody here safely. And we commit the rest of the day's activities into your hands. Dear Lord, we also want to commit all travelers into your hands as they return to their various destinations. Take them back safely. We thank you. We are grateful. Take all the glory and honor in Jesus' name.
Well, there will be a cocktail for our very special guests at the University Guest House immediately after leaving here. So let us rise for recession in the very reverse order that we came in.